Right. Okay, Tirupati, thank you for giving me this opportunity. I especially thank Mustafa sir because he has given me this slot. I'm going to present in this national webinar on biotechnology for conservation of biodiversity. This is for the enrichment of degree college teachers, which is organized by Government Degree College for Women, Department of Botany, Jagatiyal. And my topic today is green, yellow, and brown biotechnology. I'll repeat: green, yellow, and brown biotechnology. Through this lecture, I wish to enlighten all the teachers that they go back to their class and use this information for enlightening the students. So shall I start? Yeah.
Hello.
Yeah. Shall I start?
Should I start?
Hello, Tirupati.
Inka kaleda?
Yes, sir. You can start, sir.
Yeah. The first slide is about
Na photos dana indla.
Mr. Bill Gates and our Honorable Prime Minister receiving a plate in which you have different colors. The statement made by Bill Gates is, I am an investor in a number of biotech companies, partly because of my incredible enthusiasm for the great innovations they will bring. So this is a statement by Mr. Bill Gates, who is the icon of world information technology. As per this statement, biotechnology is a growing component of the society. So the next slide will show you what is the definition of biotechnology. Biotechnology's definition is, it's the application of science and technology to biological organisms for industrial production of biomolecules without disturbing the environment that improves quality of human life. So this is the definition. The application is science and technology is being applied to biological organisms, which includes plants, animals, etc. And then why we do this is for industrial production of biomolecules, which are important for human welfare, without disturbing the environment. This is the definition of the word biotechnology. Next slide shows you that there are 10 colors in biotechnology. That means there are 10 branches of biotechnology. These are in the welfare of science, development, and humankind. Red biotechnology pertains to medical and health and diagnostics. It specializes in biomedicine and biopharmacy. The second color of biotechnology is white biotechnology, which is industrial biotechnology. Production and processing of chemicals takes place there. The third one is green biotechnology, that is the topic of our today, that is agriculture. How we use biotechnology tools for crop improvement, that is going to be presented. Then we have gold biotechnology. This is related to bioinformatics. Blue biotechnology is related to aquaculture. Yellow biotechnology is related to food and nutrition. Brown biotechnology is related to arid zone and deserts, where we have crop and animal management. Gray biotechnology is pertaining to environment and ecosystem or ecology, and it deals with pollution, climate change, etc. And then purple biotechnology is very important, is intellectual property rights, where we have patents and royalties included that. The last one and the most important is dark biotechnology. We call it as biological weapons, bioterrorism, biowarfare, all these things are there. So today, my lecture pertains to green biotechnology, one. The second one is the yellow biotechnology. The third one is brown biotechnology. Are you able to hear me? Hello?
Hi, yes, sir.
Are you able to hear me?
Yes, sir, hearing.
Right, thank you.
Yes, sir, we are hearing.
The first one is green biotechnology. I have put a
You
photo
can proceed.
which I have personally taken in my own house. This is mentha leaves that we use. Okay, so green biotechnology, right? In green biotechnology, in biodiversity, I am focusing on conservation of germplasm. I'll repeat. In green biotechnology, my presentation focuses on conservation of germplasm. Right. Now the question is, what is germplasm? Germplasm is the genetic resource, the genetic material of all biological organisms, including plants, animals, humans, microbes, etc. All these things are included. That is called germplasm. What is the use of germplasm? We should conserve germplasm. We should preserve germplasm. Why should we preserve germplasm? Is we need it for food security. We need it for animal and plant breeding program. We need it for future use. So this is the conservation of germplasm. We definitely have to conserve whatever germplasm is available with us. So this germplasm is very, very important. So it is the genetic resource of all biological organisms. Right. Why do we conserve? First is biodiversity for human welfare, genetic diversity for crop improvement, species protection from extinction and endangering. We have aesthetic value for all the germplasm because of the natural ecosystem, and there is a healthy interaction between the ecosystem components. Global warming and climate change are what you call as ameliorated by the germplasm that is available for us, and then we also use this for future research programs. Yeah, there are some 4 participants have raised their hands. Yes, what is the question? Hello.
मोबाइल है जिसे रिचार्ज करना पड़ता है ना.
Pardon? 4 participants have raised their hands, please. Okay, right, we'll continue. Okay, right. Now, how do you conserve germplasm? How do you conserve this genetic material in form of seeds, in form of tissues, in form of pollen? So we have 2 methods of conservation. One is in situ, the other is ex situ. My topic does not touch with in situ. I am preferring only with ex situ. What is ex situ conservation? Outside the site, conservation from their natural habitats is called ex situ conservation. In ex situ conservation, we have a list of things like seed storage. Seeds have to be conserved. Gene bank, field gene banks have to be conserved. We have in vitro storages, that's called cryo cold storages. We have tissue culture storage. We have pollen storages. We have genetic material storages. We have cryopreservation. I'm not going to touch arboretum. So some of these I will be touching in my lecture. I will not be looking into in situ, right? So ex situ conservation. First is seed gene bank. In seed gene bank, we store seeds, right? When you store seeds, it means you're storing genes. All plants do not produce seeds. Take, for example, your rose plant does not produce seed at all. Take, for example, banana, it does not produce seed at all. So in that case, we store the other material. So all these put together, you call them as genetic material or gene bank. Okay. The seeds of food crops are stored. Seeds of rare plants are stored. Seeds of different kinds of plant species are stored. And these seeds first have to be reduced in terms of moisture content, and it is to be about 50%, right? Now, every 1 year or 2 years, you are supposed to get back the seeds and germinate them and grow them into a plant. So this is called seed gene bank. Okay, right? I can show you some pictures. I am showing you the maize seed banks. You can see seeds of maize here and then cobs here. Hundreds and thousands of varieties strains or all these things are stored here. This is rice, our staple food. India's staple food is rice. Have you ever seen this color of rice? Hello?
No, sir.
No. So you have so many colors of rice.
So nothing.
Yeah, we say that at least 2,000 wild varieties of rice are there, and they are rich in different minerals. Like what one we eat is pure white, more polishing, but it's not like that. You have so many colors of rice. You call them as germplasm. These are traditional rice paddy varieties that we grow in our country and elsewhere. And then you have seeds of gram, Cicer arietinum, cucurbitaceae, and potato. Potato is so many colors are there. So all these are called as germplasm. There are so many seeds, right? Mango is one of the important fruit crop of our country, fried fruit of our country. You have so many varieties in mango. You have so many varieties in banana. Red color banana is there. You have yellow color banana, green color banana. You have edible banana, you have vegetable banana. So you have a host of these varieties available in our country and elsewhere. They have to be preserved for future use, for replenishing our breeding system. Okay. I'm also showing you goat diversity. You know, in our country, goats and sheep are very, very important for us. They are very important for us. You have so many varieties of goat. So these have to be conserved. How do you conserve? I'll tell you the next slide. Okay, right. This is a cryopreserved seed. The cryopreserved seed can come back to germination and then they develop into seedling, right? The second kind of bank is called tissue bank.
Tissue bank.
In tissue bank, you store tissues. What are the tissues that you store? You store protocoms. You can store meristems. I'll repeat, you can store protocoms. You can store meristems. These meristems and protocoms can be put into in vitro system and then you get regeneration. They can be cultured on tissue culture medium and then you get different plants from that particular thing. Why we use tissue bank is for seedless plant. Seeds, there are no seeds in plants. So there you have to preserve them. How do you preserve them? We preserve them in form of tissue, right? So seedless plants are preserved through tissue banks. Tissue bank is one of the very important area that is being worked out world over, right? Pollen grain, right? One very interesting thing with pollen grain is we teach our BSc students that once a microspore develops into pollen grain is liberated into the atmosphere. It falls on the stigma and then pollination takes place. That is its fate. But in tissue culture, in this method, you can produce haploid plantlets from pollen grains. You can produce microspores from pollen grains. This is the beauty of this conservation method. So we can use the pollen. We can also collect the pollen of different plant species, store them, and whenever we need them, we can produce what you call as haploid plants. This is one very important thing. So pollen grains can be preserved, right? One question is, how many years you can preserve pollen grain? Every 2 years, we have to get back the pollen grain, germinate them, and develop haploid. So this is going to be a very strenuous task, but this is going to be a good source of material for future use. So pollen grain. Okay, I have put them into one flow chart. You have anthers and pollen, you have meristem, you have the material here, you have callus, you have somatic embryoids, suspension cultures. All these things are tissues which are useful for regeneration, and we can get plantlets from them, and thereby you will be able to conserve these plants. Okay, right. Where do you conserve them? In cryo banks. What is cryo bank? Cryo bank is a place where you store these germplasm under cold conditions. In liquid nitrogen, we store them, right? So all the tissues can be stored in liquid nitrogen for a very long period of time. Seeds can be stored, and then seeds, embryos can be stored, meristems can be stored. So this is one very important method of cryopreservation, right? Not only plant system, you can also store embryos of different types of animals. Not very specifically human embryo, You can also store embryos, right? Embryos can be stored. Stem cells can be stored, and these stem cells can be used in future. These stem cell embryos can be used in future, right? So you can store embryos, you can store oocytes, you can store ovaries, any type of animal tissue that can be stored and it can be used. Eggs also can be cryopreserved, right? You can store gametes also. Preserve gametes also. Gametes can be collected from different sources. Animal system, you can collect them and then you can store them in a cryopreservation at -196 degrees centigrade, right? Now we have come to a very important issue faced by our country. This is the pride of our country. Tiger is the animal of our country, national animal of our country. This is becoming endangered. People are killing this for different purposes. But what biotechnology says is you can sequence its genome. You can collect its genetic material, like eggs, whatever tissue you need, you can collect them. And then you can also go for cloning. In future, maybe we'll be interested in developing the clones. So Tiger project was taken up by CCMB Hyderabad. Professor Lalji Singh has worked on this, and now we are able to grow the number of tiger in our country. This is one very important contribution because of conservation, right? I can show you a list, right? These are tigers, these are vultures, okay? This is your sparrow. Every day we used to see a sparrow in my childhood. Even in urban areas, I used to see a sparrow, male, female sparrow. Today, I don't see them. Today, I don't see them. Okay, this is rhino. This is bustard. I'll tell you the details of this. This is black panther, visible only in our country. So they have to be protected. They have to be conserved. Their genetic material has to be preserved somewhere. Otherwise, in future, they become extinct. You're not going to get them, right? This is the deer forest lion, Asiatic lion. In our country, the number is increasing gradually. We are now protecting this is conserved. This is an animal which is conserved. We need to conserve animals. Okay, this is the beauty of biotechnology, right? Okay, using cryopreservation, we have been able to take cell suspensions, callus, protoplast, meristers, all these material of these plants, and they are cryopreserved. So whenever you want them, you want to do some research, you want to take some material, you can collect this material and then use it for regeneration experiments, right? So we are also able to regenerate from that, right? Different plants, beautiful picture. You have another research. This is from cell suspension cultures. You can regenerate a plant. This is from somatic embryos. You can regenerate a plant. Okay, plants can be regenerated. And this is a tissue culture plant. You can get plants from axillary buds. Okay, so this is one of the very important area where you can regenerate a plant which is stored in cryopreserved condition. Okay, right. You have large number of world banks, cryo banks. National Bureau of Plant Genetic Resources stores large number of cryo bank, a large number of material into that, seeds, fruits, etc. Agroforestry material, industrial plants, pollen, dormant embryos, dormant aromatic, all these things can be stored in NBPGR. Yeah, this is the NBPGR station in Delhi. You have Kuldeep Singh, who is the director. Here is Jiten Singh Sandhu is another director. They are preserving a large number of seeds in our country. Okay, yeah, this lady is called Dr. Veena Gupta. She is preserving a large number of seeds, 4,38,000 seeds and accessions. She is present. She is doing a wonderful job. Okay, then cryopreservation of the animal material. You might ask me how you can preserve a tiger, how can you preserve a lion, how can you preserve birds? You can collect the material of them. You can collect the semen, you can collect the embryos, you can collect the spermatozoa, or any different parts and tissues of plants, of animals, of fishes, birds, mammals. All these things can be cryopreserved and they can be used for further experiments in cloning, right? Yeah, have you ever heard of this animal? Anybody? Anybody?
Yes, sir.
Yeah, this is called Ongol bull. #1 world's heaviest bull. This is very, very important. It is found only in India, only in Andhra Pradesh, only in our country. This has to be preserved. How do you preserve? The semen, its material is cryopreserved in this particular cryo can cylinders or containers. So we need to conserve them. Biotechnological tools have to be applied to conserve this animal from being endangered. Okay, right. Now the next thing that we study is there are a lot of endangered plants and animals. I'll show you the list of them. These are the endangered plants. Like you have Amanotaxus, Actinodaphne, and then you have Pyrospermum, you have Gomphrena caryata, right? These are endangered. These are critically endangered also is there. Like you see Cycas bedomi is critically endangered. Okay, I have worked in my research lab on Pterocarpus marsupium. Through tissue culture, I have propagated this particular plant. Even without seeds, I can get this plant into the field condition. So these critically endangered or endangered plants have to be at any cost protected. It has to be saved. Otherwise, we are going to do rich biodiverse plants and animals, right? Okay. You should thank Professor Sureka Madam, because when she was the head of the department, she took a lead to invite this person. You see this person who is picking up seeds from the floor. He is a Padma Shri awardee. What is his contribution? His name is Darapalli Ramaya from Khammam district. What is his contribution?
Vanaji Ramaya, sir.
Yes.
Vanaji Ramaya is collecting
Ramaya,
seeds.
He has
Yes, sir.
nurtured
He is
1
collecting
crore plants. Yes, you should take his example for protecting our material, our plant material, right? So this was one very important occasion in biodiversity conservation. See, he is an ordinary man. He's picking up the seeds, these seeds that have fallen on the road. We need to inculcate this idea in the students. Seeds will become endangered in course of time. Plants will become endangered. Where are we going to get the seeds? Right? See, this is a tribal hamlet area where they have preserved so many seeds. And these seeds are drought resistant seeds, not ordinary seeds. Large types of seeds are being protected by these tribal people. Okay, right. These are the tubers. These are Adivasis who have been protecting and conserving these particular tubers, right? Okay. Now we will have some change here. You heard of Chipko movement?
Sundar Bahuguna,
Yes, sir.
sir.
Bahuguna. What is his
Sundarlal
concept?
Bahuguna.
Yeah, don't cut trees. Don't cut trees. Then he has developed a system where
The trees.
go and embrace a tree. You cut the tree, you cut more. Pardon? Yeah, so Chipko movement is one very important movement, right? Now you might say, sir, how do I protect this? The plant is coming in the center of the road widening. So how do I protect? Right, there are concepts like these. You can replant 20, 30, 40 years old plant also. You can treat these plants like this. I'm showing you some picture. You can replant a 20 years old plant from one location to another location. This technology has to come in our country. This is already available elsewhere. This technology is going to play a very important role in protecting our 40 years old tree, 40 years old neem tree, 40 years old people tree. What are we going to do? We are going to cut down. No, you are now supposed to develop this kind of culture in our students, educate them. This is the need. Each tree is important for us. It protects us from all other vagaries of the climate. Okay, right. This is another very important bird called the Great Indian Bustard. Only in Rajasthan it is available. Now it has become endangered. We need to conserve these plants. How to protect these plants? We have some biotechnological tools. Right? Next is DNA footprinting. DNA footprinting is the method of investigating the sequence specific of DNA protein in vitro. DNA binding proteins in vitro. This technique is used for DNA interaction outside the cell and inside the cell. Well, I will not go into the technicalities. You get a footprint. You get a footprint of DNA binding protein in vitro of a particular system. So DNA footprinting is going to be a very important role in conserving our plants, right? This is how it happens. This is, you have DNA here, and then you have a protein here. So you get a footprint, you get a printing. That is what we call as DNA footprinting, right? Okay. So far I have dealt with green biotechnology. Now I'm going to deal with the second aspect called yellow biotechnology. What you see on the picture is those beautiful flowers with insects like bees, etc. Okay, right. Insect farming like honey, pheromones, recombinant insect silk fiber, which are used for bulletproof vests, films, and hydrogel. This is what the outcome is. This is pheromone trap. I think everybody are aware of this pheromone trap. The male attracts the female, and then you get some kind of what you call as aroma. So it is attracted. So pheromone traps. This is there inside the insect biology system, right? Pheromone traps. These are the Indian honeybees. Save Indian honeybees. There are foreign honeybees which are killing our Indian honeybees. This has to be protected. Why you should protect them? They provide us with honey. They provide us with the sweet honey, which does not contain calorie. One very important area, protect our honeybees, called as Apis cerana indica. And then, as I've told you, using recombinant DNA technology, you can produce what you call as silk. This is the fiber of a silk, and then it can be used for producing bulletproof vests also. These are bulletproof vests also. Very important area is your recombinant insect silk for fibers. Okay, right. And then we also have hydrogels, that is natural polymers and synthetic polymers. We have nanomaterial. It can be used for drug delivery. It can be used for biosensors. It can be used for tissue engineering. It can be used for bioassay. It can be used for human treatment also. So functional hydrogels are also there in our system. Only thing is, through biotechnological procedures, we are supposed to isolate them and use them for human welfare. Okay, right. The third one and the last one is brown biotechnology. What I show you is a drought situation in a what you call a setting which is called as sand dunes, such a situation like arid situation, or you have sand dunes and the plants don't grow there. This is called biotechnology, right? In brown biotechnology, crop resistant to hot and dry conditions, that is drought conditions, crops resistant to salt, high salinity soils. There are oil-eating bugs, oil-eating bacteria, which are used to remove oils. I'll show you one by one, right? This is called, the plant that you see this here is called resurrection plant. What is a resurrection plant? This is simply a pteridophyte, Telaginella lepidophylla. The beauty of this is during dry season, it is brown. But in wet season, once you get water, 4 hours, it becomes green. How is it able to survive drought? In summer, there are genes in it which help it to overcome. What scientists are now trying to do is transfer these genes into crop plants. So genes from resurrection plants are being transferred into crop plants to develop drought-resistant plants. 33 drought-tolerant crops have been developed using this technology, right? And then transgenes from halophytes, right? This is another area. See, you see here, this is salt area, right? This is salt area. All salt is spread. No plant grows there. These are halophytes. What are halophytes? They grow in brackish water, saline water. They can survive, they can grow, like your rhizophora. Okay, they can grow. So using these genes from them, it is possible to develop plants, crop plants that are resistant to salinity conditions. So genetic engineering also can be used for developing or conserving plants. Okay, right. The important one is the super bug. This is Professor Anand Chakravarti. He developed Pseudomonas putida. he has engineered it and developed a genetically engineered Pseudomonas putida. What is the work of this putida? It will gobble up octane, camphor, naphthalene, petroleum products, anything it will start to eat. It will start to eat. Okay. In the world, his is the first biological strain which was given patent. He's given patent, right? This is the situation that we need to understand. How do you get oil to our country? Through these bigger vessels. In such a situation, what happened is this collided with the another ship, and the oil spilled into the seawater. What happens if oil spills in seawater? Birds die. The tortoise die. All the fishes die because there is no aeration. This is the situation. So what he did is he said that I am going to produce one variety or one strain of Pseudomonas that will gobble up everything. This is what has happened. Okay. So in this biotechnology era also, you can develop these kinds of system. Explain to the students that this is the need of the hour, right? Okay. In this case, you see here Bacillus pasteurii. Bacillus pasteurii is used here as a concrete. This was developed by Magnus Larsson, a Swedish scientist. Yeah, I can show you how he has done it, right? You have sand. You have sand here. You can see this, right? Sand, bacteria, and urine, they form bricks. These are the bricks. These are the bricks. They have started developing this. They have used bricks from Bacillus pasteurii. So Bacillus pasteurii is now used to develop bricks. So this is one of the greatest change that we are now witnessing in the world, that normal things can be used. So I am very grateful to all of you. Especially I wish to acknowledge today's webinar host and moderator, Padala Tirupati, Assistant Professor of Botany, and also webinar chairman, advisor, Mr. M.A. Rahman, Assistant Professor of History. I acknowledge everybody. Why? Because you have made it possible for us to present what we have in our research, in our experiments, the studies that we make, and we wish that all the students that you are going to teach in future will have some kind of impact. They will learn something, 1-2 points from this. And finally, I close with this particular slide. The best of the brain in the nation may be found in the last bench of the classroom. The best brains of the nation may be found on the last bench of the classroom. Thank you very much.
Sir, good afternoon, sir.
Yeah, please.
Sir, you started your, it is a very good presentation.
Thank you so much.
You have started your presentation, the green one with the household, you know, the mentos.
Yeah, I grow it in my house every day.
Yeah. But I just want you to request you to mention few words, what kind of research you have done on.
Yeah, basically, Mustafa sir, me, we are into plant tissue culture and genetic engineering. So Mustafa sir is going to present next lecture, wonderful lecture. He will be presenting on tissue culture, how beautiful tissue culture is working for conservation.
Okay, sir. Thank you.
Hello.
Sir.
Yeah, any more questions?
No, sir, as a whole, it is quite informative, and it's an advanced dealing with the
my earnest request to every participant is, we are in rainy season now. Teach our students to plant one tree, one sapling, and nurture it. One sapling. 99 people are attending our class. Each 99 people, when they go to their village place, one tree is going to save our nation. Don't cut trees. Cut branches, don't cut trees.
Sure.
If you want to cut trees, you have to have technology. retransplanting technology, which is very important for us. Now, I'll tell you the best example. What are we doing with the COVID-19 now? You don't have medicine, you don't have vaccine. What are we going to do? We are now facing this problem only, maybe there are several reasons. From a botany point of view, it is only because we are deforesting plants. Deforestation. Lot of plants, 100-year-old plants, 50-year-old plants are cut down mercilessly. This is the reason why our nation is facing this kind of pandemic.
Sir, I have one doubt. Hello.
Yeah, please, Chapan.
Sir, myself is Gulab, sir, your student, sir.
Okay, Gulab, how are you?
Fine, sir. I am in Kashmir, sir. Sir, my
Right.
question, one question is,
Yeah,
we see that certain plants are found only in specific regions. We call them as endemic, right?
endemic.
Sir, whether government can initiate any program to
Yeah.
conserve these species. Yesterday, I have seen in news that snake park was established in Telangana. Similarly, in a specific region, say in Khammam,
Yeah.
we found certain plant species which is found only in that region.
Yes,
Even in ADILABAD also, we found 3 to 4 species. It is never found anywhere in the world.
yes, very
So
good. Yes.
can we make the government some suggestion
Yeah.
to make certain conservation parks so
Yeah.
that those plants may be protected?
Yes, you can make, as you are working in Central Institute, you can make one to Telangana government, you can send a proposal, you can give it to, for example, our degree college is there. They can become the curators of that particular area. Biodiversity is there, not in Warangal. It is present in Jagityal, it is present in Adilabad. Yes, that can be done. I think all the degree college teachers have to think now. They can submit small projects to protect these plants. Yes, what Gulab says is right. We need to protect them. And they're not found elsewhere. See, I've been to Germany. You know what they are doing in Germany? In Munich? They are growing toddy palm trees in their country in greenhouses. Toddy palm does not grow. 6 months, there is no growth there. There is only ice, snow. They have taken toddy palms from our country and they are growing in their own country. This is the beauty of conservation.
Why? Because certain people has to depend on certain plant species. We know in
Yes,
Adilabad,
yes,
tribal people, they are depending on plants. Even
Yes.
in our region also, some people are depending on toddy plants and etc. But day by day, the number of plants, these plants are going on decreasing.
we need to
Why
work on
can't the
that.
government take some initiative to protect these
Yes,
plants
yes.
and provide employment or daily security to those poor people?
Yeah, good Gulab. We should initiate some program on that line. Very nice. Yes.
Thank you, sir.
Tirupati, over to you.
సార్ తిరుపతి సార్ మ్యూట్ లో ఉన్నారు.
Uh-huh.
Sir, one of my suggestions is
Yeah.
we can request the Department of Botany faculty members
Yeah.
to write the minor and major research projects
Yes.
to see that such rare plants are taken.
Definitely. We can do that. Minor research projects we can apply. Yes, we will
Yes.
help you. No problem.
Yes.
We are there.
Thank you.
Sureka madam is there. Professor Mustafa sir is there. We can definitely help you and prepare a small project, submit it to CERO, that is UGC in UGC place. We can definitely do that. Yes, that is also need of that.
Yes. Thank you.
Tirupati, okay?
Sir. Okay,
Thank you,
sir.
Tirupati.
Sir,
Yeah. Chappannamma.
my name is Vishnu Priya, sir.
Yeah.
I have seen in my area,
Yeah.
100 years old trees
Yes.
were cut down, neem trees.
Yes.
It used to be very helpful for us during summer time and very good breeze they used to give.
Yeah.
Just because wires are coming or construction purpose.
Yes.
I've seen in many places in Hyderabad.
Yeah.
And children are being forced to do the afforestation. One day they go and just plant and come off. What
That's
is the
all.
use, sir?
No, it should be done on a regular basis. That's
They
what I'm
dry
saying.
and there's no use after that.
No, that's what I'm saying. See, government is doing its business. Don't worry about that. What I need to say is let students learn that first. Our
Yes.
students are there. Teach them.
Regularly, if they take care, that becomes a habit, no?
Yeah, it should become our culture.
Yes.
Planting tree in rainy season should become our culture. Classroom lo kaadu manam కూర్చోవాల్సింది ఇంకా. అసలు మన చుట్టూ వాతావరణం ప్రొటెక్ట్ చేయకపోతే క్లాస్ రూమ్ ఎక్కడమ్మా? మన చుట్టూ ఉన్న వృక్షాలను 100 సంవత్సరాలు 50 సంవత్సరాలు వృక్షాలు
బ్యూటిఫుల్ ట్రీస్ సార్
చేస్తుంటే ఏం మనం సో ఆల్సో సి ఇకో మూవ్మెంట్ ఎంత బలమైన మూవ్మెంట్ కానీ దాన్ని స్నాప్ చేసి పడేసి
medicinal plants they are cutting off. What is the use? Now they are suffering.
Done. We should protest that. We should say no. This is not right. This is not right. This is not right. We should protect our environment. మన వాతావరణం నేను చెప్తున్నాను కదమ్మా COVID-19 ఎందుకో రాలేదు. ఒక్క కారణం దానికి ఆల్ ద ఫారెస్ట్ హావ్ బీన్ కట్ డౌన్. రోడ్డు పొంటి ఉన్నటువంటి 100 సంవత్సరాలు 50 సంవత్సరాల మొక్కలు వృక్షాలు అన్నిటినీ కట్ చేసేసిండ్రు. చాలా ప్రాజెక్ట్స్ కి కావాల్సిన బిల్డింగ్స్ కావాల్సిన ల్యాండ్ కావాల్సే చెట్లు కట్ చేసిండ్రు. One reason, maybe there are several other reasons. I'm not denying that, but one reason is మన చుట్టూ ఉన్న వృక్షాలను ప్రొటెక్ట్ చేసుకునేటటువంటి పరిస్థితి మన దేశంలో వస్తేనే మనం ఇటువంటి వాటిని ఓవర్కమ్ చేయగలుగుతాం, లేకపోతే చేయలేం.
గుడ్ మార్నింగ్ సార్.
ఎనీ క్వశ్చన్?
సార్ గుడ్ మార్నింగ్ సార్.
గుడ్ మార్నింగ్ శ్రీ, చెప్పండి.
ఐఎమ్ డాక్టర్ మురుగన్ సార్ ఫ్రమ్ కాళేశ్వరి కాలేజ్ శివగాసి తమిళనాడు సార్.
యా ప్లీజ్.
Sir, just one clarification and my one doubt is there, sir.
Yeah.
Nowadays, some of the urban areas, they were doing for growing terrace gardens. And then nowadays, the students level awareness created for a seed bond. Seed bond means mixing with sand with certain seeds.
Yeah.
I mean, locally growing seeds.
Yeah.
These two techniques and methods are, is it right method to develop our biodiversity, sir, in future?
Yeah, first thing is, it is the only seed.
Yes, sir.
Which is perfect. I
Thank
have
you,
explained you something about biotechnology, tissue culture, all. But basic thing is, you cannot deviate from seed. Without seed, without tuber, without sucker, without these material which are natural, you cannot improve biodiversity. What I am trying to tell you is you can propagate them, you can produce a large number, that's okay. But protect the seeds, protect the neem seeds.
Yes, it is.
Very, very important. Seed is the only method of propagation, proper method of propagation. So seeds have to be protected and that method is correct.
Okay, sir. Okay, sir. I am satisfied, sir. Thank you, sir. Thank you, sir.
Thank you so much. Thank you so much.
Thank you.
Tirupati.
Okay, thank you very much, sir.
Yeah.
And we are very enlightened, sir. We are very enlightened your session, sir. I would like to introduce your brief biodata, sir. Please accept me because of before starting your session, I am unable to give your brief biodata.
Don't worry about my Tirupati. Don't worry about
in Arts and Science College, Warangal.
Don't worry about
MSc,
about biodata.
MSc from Kakatiya University, PhD degree under the supervision of Subhash sir and research associate from Delhi University. And sir has completed FDP from Germany, Munich. And earlier, sir was worked in Krushi Vijnana Kendra, Jammikunta. And formerly served in Kakatiya University in different positions. Head of the department, Kakatiya University, Warangal. Additional controller of exams, PG and professionals, Kakatiya University, Warangal. And SEFRAC, director of SEFRAC, Kakatiya University, Warangal. And he awarded as a his students awarded PhD and MPhil 1 and pursuing 8 PhD students under his supervision. I am also one of the research scholar under My professor, Christopher Reuben, sir, and his research publications more than 60, book of authors more than 10, and teaching experience 25 years in UG and 15 years in PG, research experience 30 years, and his research field, molecular biology and biotechnology. Now he is working as a professor, Chaitanya Deemed University, Warangal district. And sir was enlightened and elaborate in the field of biotechnology applications. We are very happy and thankful to you. We hope all the participants are enlightened your session, sir, and very busy schedule. You are giving an enormous and excellent session. Thank you very much, sir, and thank you. Now I am going to introduce third speaker, our eminent professor, Dr. Muhammad Mustafa sir, Associate Professor, Kakatiya University, Warangal, and
Mustafa sir,
from SRBGNR Khammam, MSc Botany in 1988, Kakatiya University, Warangal. He was a gold medalist and a PhD on tissue culture studies in Kukurbidaisi in 1992. Teaching experience 28 years in UG and 15 years in PG. Number of PhD and MPhil awardees, 6 PhDs and 1 MPhil. 6 students now pursuing as a PhD fellows. Field of research, molecular taxonomy and plant tissue culture. Research paper published in reputed journals, books published in 6, fellow of Botanical Society, fellow of Indian Botanical Society, fellow of plant tissue culture, Indian chapter. Now is a board of studies member of Usmania University, various universities, OU, KU, Mahatma Gandhi University, and SVU, and Telangana University, SKU, and also some of the autonomous colleges, SRBGNR, Khammam, and autonomous college, Siddipet. Now, review for many journals like Vigito and BioLife, and in charge of Avenue Plantation, Kakatiya University, Warangal, last 10 years. Now I would like to invite Dr. Mustafa sir. Please give your lecture. Okay. Phone Christopher. Sir, Christopher sir. Sir. Hey, lift Christopher.
Sir, unmute. Unmute.
Sir.
Sir.
Unmute lo unnar sir.
Phone Christopher sir phone cheste idi share తీసేస్తారు.
Sir, next, good morning, sir. Next class, ever, sir.
Okay, sir. Please wait for 2 minutes. Our esteemed professor, Dr. Muhammad Mustafa sir, is ready to come on, ready to give lecture, sir. Please wait for 2 minutes.
Okay, thank you, sir. Thank you. Thank you, sir. Thank you. Thank you.